Hi, everyone, and welcome to the 2023 showcase for Next Generation CRISPR. We're really excited to hear from all the interns, and I'm going to go ahead and turn it over to the 2023 showcase for Next Gen. I'm going to go ahead and turn it on over to Michael Friend, the deep dive lead for Next Generation CRISPR, to tell us all about the internship for this year.
Absolutely. Thank you, Monica. Hello, everyone out there. I'm Michael Friend, and I'm the founder of the Minority Coalition for Precision Medicine. And as an organization, we focus on emerging technologies such as CRISPR, and we really focus on engaging communities, particularly underserved communities, around these technologies, around conversations, around the all types of implications as it relates to CRISPR. We talk about the ethical benefits of it, or sometimes the challenges, the political, the social. We even talk about the commercial, we talk about the cost. And so what you're going to hear from the interns, you're going to learn about an incredible event that they've been working on, a career day event that is looking to engage many of the high schools in the state of California. And you'll hear about that event, as well as you'll hear from each intern on their experiences and what they've learned about CRISPR and some of the companies that they've researched as well. So I'm going to turn it over to Madeline for now, and I believe Jasmine is up next. But Madeline, I'll turn it over to you and set the order. Thank you, everyone. Thank you for joining us.
Yes, thank you everyone for coming. We're really excited to just present what we've been working on these past few weeks. I know it's been my personal pleasure to get to work with a bunch of my team, and I'm really excited to present this to you guys. And Jasmine, if you'd like to start off the presentation.
All right, hello everyone. Welcome to the Next Generation CRISPR Deep Dive Showcase. This will give you an idea of everything me and my fellow interns have been working on over the past 7 weeks. The main theme was how diversity drives CRISPR innovation. So aside from creating this amazing virtual career day, we also researched, each of us interns, a different facet of how this diversity will shape up-and-coming CRISPR. Next slide. All right, my topic was on diverse perspectives, shaping CRISPR regulations and policy. Next slide. So hi, I'm Jasmine. I'm a junior attending Davis Senior High School. I'm also one of the team leads, so my main responsibilities were communicating with the interns and checking our assigned tasks and how everyone was progressing on that. I was also working heavily on creating our website, as well as networking with experts and professionals in the field and companies in order to get them to showcase their amazing companies at our CRISPR Virtual Career Day. Next slide. So while I was reaching out to these companies, mainly at the beginning of this internship, I did some in-depth research, and one of the companies that immediately stood out to me was CRISPR QC. They are really cool because they've developed a CRISPR chip and a data analytics platform. You can kind of see it outlined in green on the right. It's a small chip, and you take a DNA sample and you place it onto the chip, and then it will measure the interactions between the proteins and the nucleic acids. It will then analyze and provide feedback on how you can improve the experiment results and show you the most effective, like guide RNAs and all the cool proteins like that. Next slide. The second one was Clade Therapeutics. Clade Therapeutics utilizes a cloaking technology. The best way I can describe that is when you take the sample from a donor and then you try and insert it into the recipient. What's going to end up happening is that the recipient may want to start attacking those cells because they seem foreign. So this cloaking technology ensures that the cells can do their best work under the best conditions, and they produce multiple different types of immune system cells. They are a recently new company based in Massachusetts, and they are currently aimed at treating cancer and creating a wide range of products that are really accessible to as many people as possible. Next slide. So there are over 100 CRISPR companies right now. In 2022, the sector was worth around $3.4 billion. And by 2027, it is about to increase to about $7.3 billion. That is a 22.3% increase, which is huge. It was only in 2002 that we first gave a name to what CRISPR is. So like any new technology, we don't have a lot of current regulations. There are no ethical or international standards for CRISPR. While certain countries have specific regulations, for example, China, they require national approval for any CRISPR use. Worldwide collaboration is really necessary, though, if we want to continue to use CRISPR in an environment where it is not abused. For example, CRISPR can be used as a bioweapon, or it can be used to enhance like future genetic babies. So we really need to create these ethical standpoints in order to ensure that it is used in a way that is mainly beneficial for those who need it. Next slide, please. Currently in the US, there are no restrictions on this technology. So that means companies can experiment with it as long as they follow basic ethical considerations. But in order to run any clinical trials, the FDA must approve it on a case-by-case basis. If these clinical trials are successful, then the drug is marketed commercially. So far, the FDA has only approved somatic gene therapies. So they've not approved any germline cell therapies, which I think is definitely something to note, because these germline cell therapies will have so many future impacts that we cannot even begin to comprehend at the moment. Next slide. So I know that our wonderful deep dive lead, Mr. Friend, really drilled this into us, but we are really the voice right now. We are the people that CRISPR will affect in 50 years, and we will really be a deciding factor in how the biotech companies and big pharma and lawmakers decide to shape these international regulations, or even just in the US. The policies and regulations that we determine now and that we make effective will determine the future of genetic engineering. We have the choice now to decide if we want it to be accessible to all, because currently it costs $1.5 to $3 million just for a CRISPR clinical trial. And many insurances don't actually want to, eat that cost, especially with such a new therapy. So if we create policies right now that ensure that CRISPR can be accessible to all, we won't have a situation where only people with the means can get access to this potentially life-saving technology. Another thing is that diverse collaboration between everyone will also really help in building transparency, as well as educating others on all the possible benefits of CRISPR, which I will get into more on the next slide. Right, like I said before, we need collaboration between all parties, whether it be board members, stakeholders, scientists, public, or really even people who are currently suffering from some of these diseases. The best example I can give is sickle cell disease. Mr. Friend gave us the wonderful opportunity to talk to a woman who is currently suffering with sickle cell disease. And what I really took away from that is that many people do not understand how the disease can really affect you. She would rather not go to a hospital because she's often treated as an addict. And I think that if we ensure that people are fully educated, then this new treatment can be used in order to really meet the patient's need, not just on a technological standpoint, but also from ethical and societal considerations. So through being receptive to diverse perspectives, we can really pioneer an inclusive environment of genetic engineering. That was all. Thank you.
Thank you, Jasmine. For the next part of our presentation, we can go back to the slides, and this is where I will jump in a little bit and just talk about the collaboration of both the deep dive and CRISPR in general. Monica, you could help us with the slides, please. Thank you. So really quickly, I would just like to introduce myself. I am going to be a senior this fall at Downey High School. And in this deep dive, I was able to be a team leader, which was very fun getting to work with everyone. My job was mostly assigning work, making sure everyone was on task, but still getting the support they needed. A big part of this deep dive was communication, especially with different CRISPR companies, and just kind of forming to build what we're trying to go for, which is our career day. And that was kind of my role. Next slide, please. So 2 companies that specifically stood out to me out of these hundreds, this 100 pair of companies was Bluebird Bio. They are currently on their 3rd phase of their clinical trial, and they were a company that stood out mostly on compassion, but also that they were evolved enough to start their clinical trial. And Capside Biotherapeutics, they are more, I would like to categorize them as a engineering and manufacturing. They're also very new, and we can see that by that they don't have the clinical trial like Bluebird Bio does. And it's nice to compare these 2 different companies because they're both in different kind of stands, and also they take a different position on CRISPR and the technology and availability of it. Next slide, please. So the collaborative environment, this both comes into what Jasmine was talking about in CRISPR, but also in our deep dive. So like what I was saying, I was a team lead, and what a little bit of a side job I had to do was making a flyer for our career day. And you can see that on the right. And it was nice because I was able to get feedback from people I was working with, just making sure it was to the standards we needed, which was a great bonus to the deep dive itself. But also, while we were reaching out to these companies, it really, really just taught us that we need to reach out. This is kind of the field where you have to be willing to just keep talking and searching, looking for answers. And we're able to meet with 2 different scientists. Thank you to Michael. And they kind of showed us the same thing, that research is also so important, but it's so important to talk to your peers and have communications and discuss what you've learned and just bring that to the table and kind of like showcase that. And the career day hopefully is going to be able to inspire students, those who are interested, to kind of, this is kind of the next step, and it's going to be very exciting. And this is like a collaborative environment. So that's kind of a little bit what we got there. Next slide, please. And each company is different in itself. Every company had something specific about it that just made it stand out. And some other companies are specific, which we'll probably hear later. The progress that they were making wasn't exactly as fast-paced that it needed to be, so it would come and join with other companies. And this is kind of a case, especially in medicine and in technology, where you need to go to others to see their ideas, to see what is working for them versus what isn't working for them. And this goes along with the companies, but also in our deep dive and how we work together, but also in the field and how it works. And I think this is the end of my presentation, and I pass it on to Nicholas, who's our next presenter.
Skipped a couple slides there. Hello, everyone. My name is Nicholas Kuipers. I'm also an upcoming senior at Downey High School, as well as a team leader. Next slide, please. So when thinking about CRISPR, it's important to consider ethics. Next slide. And when considering ethics, it's important to remember where CRISPR is applied. Next slide. There are 3 main places where CRISPR is used, that being crops, animals, and families. Next slide. When looking at how CRISPR affects crops, it's important to remember these key bullet points, that mean genetic diversity, sustainable agriculture, marketing transparency, and equitable access. Genetic diversity is important because oftentimes things like diseases can wipe out entire crops when the crops aren't genetically diverse. However, CRISPR can also help affect these crops in a beneficial way by directly countering these possible diseases. There's also important is market transparency. Many people do not yet trust these new designs of CRISPR, and it's important to let the public know where it's being used, when it's being used, in order to make it so that in the future, uh, more people are likely to trust these new upcoming adventures. Next slide. As well as crops, there's animals. As with all clinical testing on animals, it's important to remember the animal welfare as well as the disease resistance. If an animal were to escape, a disease could potentially wipe out entire herds. But also with animals, it's important to remember novelty and misuse. As with pugs and selective breeding already, I believe it has been misused just so to kind of satisfy the recipient instead of actually helping the animal. It's important to remember what is best for the population as a whole and not just use CRISPR to change things that aren't necessary. Next slide, please. I believe the most important part of ethics is on the humans, and so we'll explore that right now. Next slide. When looking at CRISPR's effects on humans, it's important to remember two different classes, adults as well as babies, which I will get to in a moment. But with adults, it's important to break it down among diseases, cosmetics, and some potential risks that might align itself with CRISPR. As the diseases, it's practically the same, but it can be used to specifically target things like sickle cell, as stated earlier. However, with adults, it's far more likely that CRISPR is used than for cosmetics than on things such as animals, I believe. But still, it's important to remember that we don't want to eliminate the human experience and that some difference is always important. Next slide. All right, now we're going to get on to designer babies. Designer babies is a key, or it's a hot news title you might have heard of before. But designer babies uses CRISPR when the baby is an embryo to modify it in order to make it taller, more athletic, smarter, or even things like change eye color. While this might seem appealing initially, it's important to remember that doing these things can eliminate some of the human experience, and it's important that we all accept each other instead of trying to become each other. Next slide, please. But some things to remember is that innovation is coming. If we don't put these ethical guidelines into place now, someone else will, and they might not agree with the rest of the community on what is and isn't acceptable. It's important to put humanity first when designing these. We don't want mass famines wiping out because the crops were a little bit more efficient. And it's important that trust should be earned through things like marketing transparency and public opinion. These are some key things I think that should be remembered when considering the ethical impacts of CRISPR. That's it for me.
All right, seeing how it's now my turn. Hello, everyone. My name is Noah Espudo, and I will be presenting a lot of the ways that CRISPR can be used to help the world globally. And just starting off, I'm a student currently attending Warren High School as a senior. What I did during this internship was I was a project manager, and I coordinated very much with a lot of my fellow interns and peers, be it trying to coordinate with other companies as well. I know all of us were very much working to communicate with companies on LinkedIn, creating LinkedIn accounts, trying our best to obtain responses for our upcoming career day. And besides that, I worked very closely with Michael, delegating work to everybody else, as well as working on a lot of that work myself, going over a lot of the emails that would be sent to a lot of our stakeholders or presenting. And next slide, please. Right. Firstly, some of the notable companies that stood out to me throughout my research were number one, Synthigo, which is among the largest CRISPR companies in the United States. And another reason that they stood out to me was because they're among the most open and informative CRISPR companies. Throughout their entire website, they have an entire tab just dedicated to informing people about what CRISPR is and providing tools to help other people just analyze certain CRISPR aspects of CRISPR. Furthermore, they're one of the few companies that actually engages and has numerous partners and is looking to almost confederate with other companies and regulate as well, trying to come to terms with everybody and just keep everything quite ethical as well. And our second company, or the second company that stood out to me, was Sparing Vision, which is based in France, which was the only European company that I came across in my group of companies that I had to research. Furthermore, they had among the highest amount of women in their management, which was half female and then half males, which was quite interesting considering when you're going through all these companies and looking at their management, they seem to be it's a very male dominated field. And furthermore, they use CRISPR on eye diseases, which was quite different compared to everybody else who seems to be focused more on curing gut diseases or even hereditary diseases. Next slide, please. So one of the ways that CRISPR tackles or can tackle a lot of our global issues would be its effect on agriculture. It very much has the potential to ultimately solve a lot of our famine crises around the world. And in doing so, or one of the ways it does so, is by allowing for longer lasting food. Basically, what CRISPR does is it allows for people to modify the food's DNA in order to, or genes, in order to essentially make it more resilient to famine, drought, diseases, and allows it to grow in harsher conditions, which is quite revolutionary considering in a lot of areas, there's no potential to even grow food. Furthermore, it's working to make healthier foods more appealing to people, which at least in the United States can be quite massive, especially since in the United States, we have a high rate of obesity, and it's far more likely and it's far healthier for people to be more, it's far better for people to eat healthier foods as opposed to just junk food all the day. Furthermore, it also makes lab-grown meats a very feasible alternative, which can help with lots of greenhouse gas emissions. And yeah, next slide, please. CRISPR has a large effect on pharmaceuticals as well, especially hereditary diseases, though at the current moment, not many clinical trials have been conducted on people. Notably, it's been mostly conducted on animals. For instance, it has been conducted, there was a study conducted where mice were able to have their hearing restored, even though, and these mice were suffering from a mutation very similar to humans, where it causes them to be deaf at birth. And CRISPR had the potential to eliminate that in those mice, or at least greatly reduce it, which could greatly help people as a whole. Furthermore, it has experienced quite some success with SCD and beta thalassemia, or thalassemia, which was announced in the ASH 2020 conference, where I believe 3 patients with sickle cell disease were treated and have had much more success in the 1 year that they were being treated compared to all the other years that they were just having their symptoms reduced. And I believe in one of our previous meetings, it was brought up that malaria or sickle cell disease was actually favorable in certain areas where malaria was quite prevalent. And through my research, I found that that might not even be necessary if malaria isn't even a concern, which CRISPR has been used to tackle through basically mosquitoes being altered so that way, if they have the genetics that spreads malaria, if that is stymied, well then, when they are released to the population, they mate with other mosquitoes. These mosquitoes will be sterile if they do not have, or if they have malaria, which has the potential to eradicate malaria as a whole. Next slide, please. And finally, CRISPR can has a large effect on our energy crisis. Throughout but very much in the past 5 years, there's been a large concern about going electric, which might not always be the best considering a lot of the materials are non-recyclable. However, CRISPR can help with many biofuels. For instance, it's shown to reduce lignin and other inhibitors for more efficient energy. And it can also be used as to enhance substrate utilization capacity, allowing for a more efficient and cleaner burn. And it also is used to improve the metabolic flux in a solvent and make it more balanced. Again, just focusing on efficiency and making everything far more feasible and reliable. Next slide, please. And some final remarks I just wanted to leave with everybody is that CRISPR is a very young technology. It's very expensive at the current moment and has little to no regulations, which is a massive concern because that means that at least for the next 5 years, we won't even be able to see CRISPR really be used on a global scale, or even I would say a national or local scale. However, that doesn't mean it's not feasible at all. It really just means that you'll have to put more effort into it. But ultimately, once a lot of the money and the time has been put into CRISPR, it has potential to revolutionize so many things and solve so many global issues that currently, I would argue, as a human race, we cannot argue with, we cannot solve without. And all right, I'm gonna just pass it on to whoever was next.
Hello, my name is Jessica. I'm a senior at Warren High School, and in my group, I worked as an intern on researching CRISPR application in many fields that focus on the 2 topics that we're talking about today, which is agriculture and the environment. I also worked on different CRISPR companies as the rest of my co-interns did, and assisted in the operations of our event, like our future career day coming up soon. Go to the next slide. So here are the companies that stood out to me. Amendo Biotherapeutics is one that especially stood out to me because their goal is to make CRISPR fit like a wide range of diseases versus making a disease have the standard to be used by CRISPR technology. And they're really focused on tackling those challenging diseases that aren't really talked about or solved quite as yet. And then another company that stood out to me was Emerzine, which I'll talk about later in my presentation, but it's CRISPR's applications with agriculture and developing medicine. Go to the next slide. Innovations in agriculture. CRISPR technologies had many more uses that my co-interns will discuss later in our presentation, but agriculture is one that displays the versatility of this technology. In the future, CRISPR technology in the world of agriculture will have many effects on effective farming. Part of this technology coming out, genetic breeding and selecting traits have long occurred, but now using CRISPR genome editing, plants can be created to have desired traits just by altering their DNA rather than introducing foreign DNA. In this way, this technology avoids many of the side effects presented by previous methods that were used before, and instead it improves accuracy in the process. Although not perfect, as CRISPR technology is still relatively new, in the future it's very plausible that this technology can be used instead of older methods. Using CRISPR technology has helped tremendously in plant research. For example, by breaking the gene, you're able to tell with CRISPR tools, it will break a certain part of the DNA sequence and observe how those breaks affect the plant. From the experiments, they can deduct which genes exhibit which trait in plants and allow scientists to get a precise understanding of plant DNA. And with more research, they're able to utilize more CRISPR editing technology effectively in changing a plant for the better. Another way CRISPR technology is used is to improve crop resistance. I know it's been touched on before, talked about before by my fellow interns, but especially emerging consequences of climate change, can make crops more resistant to conditions like drought, salty soils, or extreme temperatures. The research on this is still developing, but has so much potential to help staple crops like wheat, rice, and and corn to be able to grow in these conditions, which will ultimately increase crop yield and humans' access to food. CRISPR can also improve crops resistance to disease and pests in a similar way with genetically modifying the crops. They can edit proteins that viruses, bacteria, and fungi or insects survive off of, so they can't take advantage of that plant. And it'll improve the overall value of one crop as it has less chances of becoming bad. CRISPR can also improve more commercial properties by preventing bruising, allowing longer shelf life, and changing the appearance or flavor. Not only will these crops look better, but they'll have better nutrition and taste when consumed. Using CRISPR may allow easier access to these desirable crops, which in turn can help those struggling to afford good, nutritious food have better access to it. And then one of my favorite and most interesting way CRISPR is used in agriculture is used to produce our industrial chemicals like vaccines, insulins, organic compounds, natural oils, and more. And then one company I researched on this was Emerzine, as I talked about before, and they're using CRISPR technology to make a non-addictive pain relief medication out of poppies with the goal of helping stop drug addiction, starting with medication. CRISPR technology may make it easier for companies to turn plants into little factories by ingesting coding DNA into them. I talked about some of the more common ways CRISPR is actually being used in agriculture, but still research is continuing and who knows what the future holds. What we know for sure is that CRISPR technology is emerging with huge potential in the farming field. You can go on to the next slide. My second topic I'll be covering today is CRISPR and environmental conservation. Alongside agricultural advances with CRISPR, this genome editing tool can be used to help conserve the environment in many ways through editing plants and animals. So researchers are looking into CRISPR technology to help reduce or reverse the effects on climate change. One way this can be done is through lowering emissions, greenhouse gas emissions. Been an issue that can destroy ecosystems since it's a main contributor to warming. CRISPR reduces effects with editing agriculture and livestock. CRISPR technology has the potential to change some of previous methods that are used. For example, developing different types of grass that cows can digest can reduce methane emissions from cows. And they're also studying microbes. That is one of the major contributors to greenhouse gases to help reduce emissions in the future. And these engineered plants can also enhance soils or plants to store carbon for a longer amount of time, which will pull carbon from the atmosphere and kind of reduce the cause of climate problems. And I mentioned it earlier, but improving crop resistance helps plants deal with the effects of climate change with salty soils, increased temperatures, and more. And then one of the effects of global warming is coral bleaching with warming waters. And in the future, CRISPR may be able to edit the algae so they have a higher resistance to these warming temperatures, which would preserve the ecosystem. And then another way that CRISPR can help with preserving biodiversity is making endangered trees or species more resilient in the future in the face of natural disasters, pests, or more. And if you can go on to the next slide. One interesting example of how CRISPR can actually be used to help wildlife is their identification with CRISPR tools. In a study led by scientists at UC Davis and California Department of Water Resources, they demonstrate how a CRISPR tool called Sherlock helped them differentiate between two fish species, one of them being endangered. And through a swab, they were able to tell which fish is which. This goes to prove how CRISPR tools can be used rather than just gene editing. And in this case, it's vital that we can identify these fish in a timely manner to address issues. So this right here is just a revolutionary study, and it shows how the future of CRISPR can help environmental monitoring in this way. Although CRISPR has so much potential in the agricultural fields, there are ethical concerns that arise with genetic editing in animals. People are afraid that if we have livestock with better traits, that being disease resistant, it'll encourage farmers to practices of over farming, just as an example as to why ethical concerns are raised. Still, most researchers and scientists agree that the future seems far for genetic editing to save endangered animals as best as they can. What's important to remember is that many of the methods I just mentioned are still under research and being developed, but aren't actively being used in many ways that I discussed. CRISPR has plenty of potential for the future, and hopefully in the next generation, we can see more of the applications of it. And thank you.
Hello, my name is Eric Rizzo. I am entering my senior year at Downey High School. I had the task of gathering knowledge from several science companies, such as brainstorming as well as organizing information during this project. Next slide. Notable organizations. One of two organizations that stood out based on their work would be Gressel Biotechnologies. Gressel Biotech, for short, is researching cell and gene therapies for cancer and autoimmune disease patients that provide fast, significant, and long-term responses. Gressel Biotech is a global clinical stage biopharmaceutical company dedicated to discovering and developing innovative and highly effective cell and gene therapies to address major industry challenges. Gressel aims to approach CAR T-cell therapies with fast T-CAR and true U-CAR technology. Another notable organization will be IceCure Medical. IceCure Medical is an Israeli medical device firm that was created in 2006 to advance cancer therapies such as tumors. A system called the ProSense system is an optimal solution for breast tumors, which allows the progression of the treatment to be much quicker. IceCure's fundamental technique is based on cryoablation, which uses below freezing temperatures liquid nitrogen to eliminate tumors effectively, rapidly, and painlessly without the need for surgery. IceCure's mission is to use their optimal solution. And that's it. Next slide. Policies and regulations. In the United States, CRISPR is legal. There are no restrictions from organizations, for example, hospitals, from utilizing CRISPR technology. Clinical experiments using CRISPR are currently being pursued by a variety of hospitals and biotech organizations. The FDA does, however, oversee their clinical trials, and if the trials are successful, the FDA will grant these organizations permission to market the products. The FDA approved somatic gene therapy, so the organization is allowed to use it on patients. The US Congress implemented a policy that prohibits the use of federal funds for human germline gene therapy research. Next slide. Benefits of using CRISPR. CRISPR can be used in various other ways other than human DNA, such as in our environment and the food we consume. Prevent endangered species from obtaining deadly illnesses. Scientists are able to help fight the rise in animal extinction around the world by utilizing CRISPR. By doing so, we can help preserve the creatures from extinction. Scientists can change the DNA of almost extinct species to prevent them from contracting a specific illness and becoming extinct. Additionally, CRISPR may someday enable researchers to produce a hybrid form of the extinct animal. Prevent pathogens. Corn, rice, mushrooms, and other common foods can benefit from CRISPR. Crops are prone to diseases including fungi, bacteria, and viruses. In order to stop diseases from infecting crops and spreading to other crops, scientists can utilize CRISPR on plants to prevent this. This allows farmers to not have to worry about infected crops and possibly an individual consuming one. Another benefit of agriculture can benefit from CRISPR would be an improvement on nutritional value and includes more kernels. CRISPR genome editing tools have many applications in plants. Researchers use them to learn more about plant biology. Plant breeders use them to create crops with the desirable traits. Scientists can even use them to turn plants into leafy manufacturing facilities. And that is that. Thank you.
Hi, my name is Alyssa Jin, and I'm an incoming senior at Downey High School. During this deep dive, I researched and contacted CRISPR-related companies, and as well as completing and contributing to tasks given by my team leaders. Next slide, please. Two companies that stood out to me were Proof Diagnostics and Primera Therapeutics. Proof Diagnostics is developing a CRISPR-based COVID-19 molecular test. They're unlocking CRISPR technology to make powerful molecular testing accessible to everyone. Using CRISPR and RT-Camp technologies, their scientific team is developing a simple, streamlined platform that can quickly test for the presence of SARS-CoV-2 virus. Their goal is to transform personalized health and expand diagnostics to detect diseases to allow people to live healthier lives. Primera Therapeutic addresses the root cause of mitochondrial diseases by developing a gene editing platform to target inherited mutant mitochondrial DNA. Their first approach uses a fully customizable tail-based system to specifically target a stretch of DNA and a molecule that can correct the mutant DNA in the mitochondrial genome. It enables precision gene editing to be deployed at the mitochondrial level. And their goal is to accelerate the development of mitochondrial disease cures and promptly aid this underserved patient population. Next slide, please. Education and outreach. Some educational programs include gene editing 360 degrees CRISPR in a box. It's from the brand Christiana Care Institute, and it aims to empower, inspire, and engage the next generation of scientists committed to advancing gene editing technology. CRISPR in a Box is a teaching toolkit that provides students with a hands-on learning experience in CRISPR gene editing. And biotechnology training and education provides CRISPR gene editing certified courses for all. They provide flexible instruction, engaging videos and activities, and practical in-demand skills, as well as CRISPR classroom bundles. They serve a diverse community of learners. And Innovative Genomics Institute, which is a global leader in CRISPR genome research, provide educational materials to make science and societal impact accessible and engaging for anyone who wants to understand it. They have resources that include What is CRISPR, CRISPR Made Simple, CRISPRpedia, Illustrated Glossary, Games and Activities, and Multimedia Library. These all serve a diverse community of learners, from students to CEOs to basically anyone who wants to know more about CRISPR. Next slide, please. And some helpful initiatives to get students interested in CRISPR and STEM-related careers include Practical Actions, which provides resources and information to find STEM-related careers based on age and subjects, which can serve a diverse community. Battelle has an article about that promotes an increase in opportunity and diversity in STEM, and the British Council has an article inspiring young people to consider a STEM career path. And these STEM career paths can lead to more researchers and scientists that can help discover more about CRISPR and what it can do. Thank you.
Okay, hi there. My name is Arman Akbari, and I'm a rising junior at Davis Senior High School. And in this deep dive, I was a team lead. So it was, what I was doing was I was making sure that everybody knew what they were doing, what tasks had to be done, the pipeline. And another thing was I was maintaining continuous workflow. We knew what we were doing and we're going to do it on time. Next slide. All right, so a few things I did in this internship. One thing was I gathered 2,800 high schools in California, which would really help us with contact information and contact them for their event, our CRISPR Career Day. Next. Another one was the union and high school districts in California, which wasn't as much as the high schools, but it was pretty significant, with also contact information. Next. And finally, the companies. This was really helpful. We would list all the LinkedIn and the companies and the company website, et cetera, the research we did on them. This would like make it more organized and so we could go back to it in future cases. Now let's get into the core things. Next. So two companies I worked the most with and really, connected the most with was Avectas was one of them. It's a company based in Ireland, and something big they have is called Soluport technology. And what this does, so there's two types of CRISPR technologies. There's in vivo and ex vivo. Both have different pros and cons. And what it does, it kind of just smushes them together and takes the pros of both of them. So it takes ex vivo editing and it combines it with in vivo pros. So I'll explain this, but in other words, there's a little container and it takes cells out of the body, but it still has the functionality of in vivo. And Avectas as a company has patents and is working on clinical trials, getting that approved. And for the career day, I worked with them and they're on board with a video instead of a live presentation because of time zones. But anyway, let's move on to Beam Therapeutics, another company. Another thing interesting about them was instead of gene editing, they're more precise into base editing, the actual nucleotides of the DNA. And they're working on in vivo and ex vivo, outside the body and inside the body. And right now they're working on medicinal therapies, curing disease, genetic diseases, etc. And how they're changing it is by a process called deamination, which is a chemical process, probably take a bit of time to explain, so I'm not going to do that, but we'll get more into that later slides. Next. And just a short timeline on the history of CRISPR. It was studied from 1987 to 2005, but really put their foot down with the CRISPR in 2005 by Francisco Mujica in a university in Spain, and discovery and the naming of it. And in the same year, they discovered the Cas9 protein and the process by Alexandre Boulatin in France. 2008, they discovered the spacer transcription to RNA, which was thought to be DNA, wasn't. Jean van der Oost in Netherlands. 2008, that same year, CRISPR acts on DNA instead of RNA, unlike they thought. 2010, a scientist in Canada discovers that Cas9 creates breaks in bacteriophage DNA to stop it from infecting the cell. And in 2011, they discovered that CRISPR is heterozygous in multiple species, so it can work in more than one species of bacteria. And finally, in 2013, they harnessed it for genome editing in other types of cells. And from that point on, it's kind of been specialized by companies adding their own types of CRISPR methods and strategies. Next slide. Now I'll focus on industry innovation. So we're going to start with therapeutics. I've already talked about Beam Therapeutics and their base pair editing, but as you can see in the diagram, they can, for example, change a C nucleotide, which is cytosine, to a U nucleotide, which is uracil. And from the diagram, you can see the chemical structure. They can change the amino acid, the ammonium at the top to an oxygen, and then the 6-carbon sugar, they can change a nitrogen to an amino. Ammonium. Sorry, I keep mixing that up. In other words, it's really precise and it really limits a lot of the probabilities of having off-target effects and genetic malfunctions. And what they're focused on doing is treating diseases, they're both at the zygote and in gut diseases or genetic diseases. Let's talk about the next company. The next company is Biohen, company in China. And what they've done is they've created a more resistant cRNA, and they've called it cat RNA. And you can see in the diagram below that unlike the normal RNA on the left, cat RNA has a 5' cap and a poly A tail. And what this does, it again makes it more resilient, more durable, less chance of off-target effects, less chance of malfunctions, and overall greater potential of editing T cells and zygotes and other human body cells. Last one is Tessera Therapeutics. And they've taken an entirely different approach. Instead of editing genes, they've used gene writing, which is a much bigger scale with multiple genes. You can see in the picture below that they have a protein and RNA, and they transcribe the RNA into DNA, which later is going to splice into the actual DNA. In other words, it's multiple genes. This is good for editing large portions of the genomes using mobile genetic elements, and is really good, again, for curing diseases that require more than one gene, or creating enzymes or proteins with this longer method. Next, please. And the second innovation is agriculture. First company is Plant Edit, and they're using CRISPR to create soy-based oil, which is really interesting, and sustainable fruits. What they're trying to do is create more sustainable produce. In other words, as climate change and so nutrition is lowering and climate change is getting worse, we're trying to counteract this by using CRISPR to edit the seeds in the plants so we have better plants and better produce. Pairwise is doing something similar. Their mission is to build a healthier world using more nutritious vegetables and fruits. And they're planning to use CRISPR to increase plant durability in climate change and to increase crop yield. An example of this, they partnered with Bayer to create corn with 20% more kernels. Now, 20% is a lot, and this will probably help a lot of countries that don't have that much agricultural, industries. For example, countries that have deserts. This will really help world hunger too, have a lot of people getting access to this nutritious food. And the last one is Inari agriculture. And what they're doing, they're also using CRISPR to edit the seeds of the cells, seeds of the plants, my bad. But something they're also doing, they're using AI along with it. They're planting plants to grow best in their original natural local temperature. And the process is, as you can see in the picture, is that they edit it using CRISPR. They edit the sequence. Then they see the performance, the plant characteristics, and they use AI along with their know-how, and they go and edit the sequence again. And this cycle will just keep repeating. And it's really more efficient using AI, obviously, it's a robot. And it creates better output and more produce yield. And overall, it's a better method of attacking the problems they have. And that's all for me. I'll hand it off to the next person.
Hi, I'm Daniel Hernandez, and I'm an incoming senior at Warren High School. During this deep dive, I utilized different platforms and services such as LinkedIn to try and contact companies for our upcoming career day. Next slide, please. So some notable companies I came across through my research was Servier and Sarepta Therapeutics. So Servier, they're a huge company with a global mission, and they have outreach all around the world. And they're mainly focused in treating various forms of different cancer, and they already have 4 drugs approved for use. Sarepta Therapeutics, they have made many strides in finding new treatments for different forms of muscle dystrophy, and they already have several drugs created that are ongoing clinical trials that will hopefully make a huge impact. Next slide, please. So I researched about important contributors to CRISPR technology. So the technology we have today allow for the many different applications of CRISPR, such as the treatment of genetic diseases and improvement in agriculture. These advancements have laid the groundwork for future improvements that will hopefully save and help many lives. This is thanks to the many people who have put their all into discovering and inventing new ways that CRISPR can be utilized. So to name a few, we have Jennifer Doudna, Feng Zhang, and Matthew Porteous. Next slide, please. So Jennifer Doudna, a biochemist from Harvard University, is the main pioneer of CRISPR technology. Her discovery that CRISPR could be utilized for modifiable gene editing is what set the groundwork for the current CRISPR technology today. This discovery was crucial as previous attempts and methods were nowhere near as efficient as utilizing CRISPR was. Her continuation of her work by developing new ways to use genome editing for CRISPR earned her the 2020 Nobel Prize in Chemistry. She has also co-founded her own company called Mammoth Biosciences, which she currently still runs to this day. Next slide, please. Feng Zhang, a biochemist from Harvard and Stanford University, has helped to majorly expand the applicable uses in CRISPR technology. He focused on trying to use eukaryotic cells instead of bacteria. Because of this, he was able to pioneer new methods of genome editing tools that could be used in eukaryotic cells that utilize natural microbial and CRISPR systems. Since then, he has made these tools widely available for others to use, which has drastically increased the efficiency of biomedical research everywhere. Now, Zhang is focusing on trying to understand brain function with the help of CRISPR. Through this, he hopes to be able to treat complex brain disorders that haven't been treatable before. And next slide. Matthew Porteous, a graduate from Harvard and Stanford University, has greatly helped in developing new therapies for genetic diseases. He's one of the pioneers who has focused on using cell-based therapies and has specialized in hematopoietic stem cell transplantation. He has also worked to use gene editing through the use of homologous recombination to try to create new therapies for genetic diseases. Porteous hopes to help those with genetic blood disorders such as hemophilia and sickle cell anemia. Currently, He's trying to better understand genetic diseases that affect children. He also works at a pediatric hospital to provide care for his patients. And that concludes my segment, so thank you for your time.
Hi, I'm Yuhan Du, but I go by Anna. I'm currently a senior at Davis Senior High School, and like everyone else, I'm part of the Behavior Group Next Generation CRISPR Summer Internship, and then we work to research on specific CRISPR companies, as well as research into deeper into our areas and fields of CRISPR. So next slide. So companies that stood out for me were Pact Pharma and Poseida Therapeutics. Pact Pharma is a privately held immuno-oncology company dedicated to engineering T-cell therapies by seeking to harness the curative potential of each patient's immune system to treat their cancer, as well as to advance into the field of immune-driven medicines. And so for Poseida Therapeutics, they are a clinical stage biopharmaceutical company whose gene editing technologies and delivery platform allows to target many different cell types, treat cancer and other rare genetic diseases. By focusing on specific cell populations, they can potentially advance their CAR-T therapies with an early memory phenotype for patients fighting cancer. So both of those were super interesting. Next. Yeah, so CRISPR applications in veterinary medicine. So CRISPR and other gene editing technologies are thought to hold the promise of developing diagnostic procedures, treatments, vaccinations, and other changes in industries as diverse as clinical medicine. So there's been studies that show that a CRISPR insertion of a gene helped produce a tuberculosis-resistant cattle, and a deletion of a CD163 gene helped produce a PRRS, porcine reproductive and respiratory syndrome resistant pig. So both of those are really groundbreaking applications of CRISPR editing in livestock. So with the use of CRISPR gene editing, scientists can quickly produce cell and animal models that can be utilized to advance the study of diseases like cancer and mental illness. Next. Yeah, so in a study in 2021 that focused on the genome editing strategies to protect livestock from viral infections, concluded that genetically modified livestock can thereby reduce disease outbreaks and decrease zoonotic threats. So that was pretty cool. And I heard this morning that UC Davis has partnered with UC Berkeley as well as UC San Francisco on a $70 million funded project to help climate change. So they're going to use the CRISPR gene editing tool on the microbes in a cow's gut to help fight and cut down climate change emissions caused by the cattle, which is an example of a current event happening in 2023 regarding animals and the use of CRISPR. Okay, next. And yeah, that's it from me.
Thank you so much to everyone. This is the end of our presentation. I really appreciate your time just to come and talk about our deep dive. And I'll hand it to Michael if he wants to finish off with any words.
Well, thank you. Thank you so much. I learned a lot about particularly the last slide around veterinarians and how CRISPR is used in that space. And so that was different. And I think what was exciting for me is how you all really went into these companies to expound upon these companies and what they're doing. And so that was exciting as well. I'm very proud of you all and the work that you've done, even particularly around Career Day, a very amount of extensive work. And so, and to this day, we're still hearing from companies. And I had a conversation, you all were a part of the conversation with Manel, who spoke to us out of Europe. And so spoke to him on today, and he's committed to continue to work with us. He's going to schedule a meeting with CRISPR Medicine News. And so, as you mentioned, some of you will more likely be interviewed as well. So I'm excited for not only what you've done now, but what's going to happen over the next couple days, weeks, and months to come. And so thank you all so much. I certainly appreciate you all. And I know there could be some questions out there, so I want to make room for anyone that may have any questions. I do know there was one video that was sent in by one of our interns that couldn't make it, and just wanted to know if we'll be able to show it. I know we're, in terms of time. And so I'm going to turn it back over to Monica at Beha to kind of field any questions that we may have, even in the chat or live. Thank you so much. Thank you, everyone.
Thank you, Michael. And thanks, everyone. I always learned so much during this. And wow, just amazing work that all of you have done. Before I open it up to any questions, and please feel free to put it into chat, or for those of you that are watching live stream on YouTube, please make sure to put that in there and we'll go ahead and answer those questions. But we have Gary Pearson here from Davis Joint Unified School District, and I wanted to give him the opportunity to say some words. And I want to also mention that the internship would not be possible without the partnership of Downey Unified School District, Glendale Unified School District, as well as Davis Joint Unified School District. So Gary, thank you so much for being here.
It's a pleasure to be here. I've been doing these deep dive presentations over the past few years. And just so you know, I'm the Career Technical Education Grant Specialist for the district, and help to fund a lot of these different activities. And it's just a lot of fun to listen and to see the growth in all of the students as they progress through this internship, and being on the ground and seeing how everyone kind of fits in, and seeing the presentations at the end is just an awesome experience.
Thank you so much. And now I'm going to go ahead and open it up to questions. So please feel free to put that into the chat, or as I mentioned, put that, for those that are watching on YouTube, put that into the chat there. I do have a question for everyone. I'm very curious, since how much did all of you know about CRISPR before starting the deep dive? Were you familiar with it at all, or was this brand new?
I guess I can answer it first.
Sorry. No, go ahead.
Okay. I knew a bit about CRISPR. I knew it was a gene editing tool, but to be honest, I didn't know that much. This internship really helped me expand my knowledge about the process and how it really works, and other considerations and thinking about it. So I knew a bit, but not that much.
Thank you.
Right. Much like Armand, I think going into the interview that we had, I did a quick Google search, just trying to familiarize myself just what CRISPR was. And after that, that was basically all the information I knew about CRISPR prior to this experience.
Thank you, Noah. Would anyone else like to comment and answer?
Yeah, just like with everyone else, I didn't know much, but I think we talked about in our meetings before that not many of us did, but Michael actually introduced a lot of us to it and educated us well on it.
Thank you, Jessica. So we have a couple of questions in the chat. Would you now consider a career in biotechnology?
I can actually answer that question a little bit. Kind of going into the internship, I was more focused on the engineering aspect, but I think doing this internship and knowing more about CRISPR, it's really like piqued my interest into biotechnology. So I'll definitely be looking at that. I think now I would 100% consider a career in it.
Thank you, Madeline. That's exciting.
I would also consider a career in it now. I've been very into robotics for like the past 6 to 7 years, but now I'm really looking into applications of how it can be combined with like AI and prosthetics, and how we can improve that sector of medicine.
Great. Thank you, Jasmine. Anyone else? All right, let's move on to the next question. What did you learn about yourself after going through the summer internship?
I found how important it is to be transparent in the issues that arise with everything, basically, and to open the conversation up to new people in order to say the issues in order to more effectively get the work done. So just, it kind of helped me to be more open about some issues that maybe before this internship I would have stayed on a week trying to figure it out on my own.
Thank you, Nicholas. Alyssa, what about you?
It taught me to be more educated and just keep an open mind about everything. Just because before this, I didn't really know what CRISPR was, and it had been going on for like a couple years. So it just opened my mind about how much information I could be missing if I don't like stay up with like the news and just all this research coming out.
That's great. Thank you, Alyssa. Daniel, what did you learn about yourself after going through the summer internship?
I learned I can get more out of my comfort zone because like I've never done anything really like this before. And so after doing all this research and learning more about CRISPR, I just feel more confident about my ability to learn new things and be able to communicate them with others.
That's great. Thank you, Daniel. And Eric, what about you? What did you learn about yourself?
I learned to be open-minded, to discover new issues that people have that can be solved by using CRISPR.
Thanks, Eric. We have one other question. After all the research you all did, how can AI be used in conjunction with CRISPR?
I think I can talk. Personally, I think that we can use a lot of AI algorithms, especially when we're like looking at RNA sequences. And I did talk about CRISPR QC, who created a CRISPR chip, and they looked at all of these different CRISPR proteins and analyzed the different interactions between these proteins and the nucleic acids and how we can maximize the efficiency, as well as like predictive outcomes and like just creating large databases of potential drug targets and how we can apply that.
Fantastic answer. You did your homework. I appreciate that.
Thank you, Jasmine. Would anyone else like to comment on that question as well?
I think I want to just say, I think it's less a matter of how we should do it or how it would be possible, and more a question of if it should be something we do.
Thank you, Noah. Are there any other questions? Just want to make sure that I'm checking all the chats. All right, I think that concludes our showcase for Next Generation CRISPR. Thank you so much to everyone, and really appreciate all of you tuning in on our YouTube channel. We do have a few more showcases coming up. So we have one in about 20 minutes at 4:30, and then we have 4 more on Monday. So I encourage all of you to please watch on YouTube as we learn on what all of our interns have done for the past 7 weeks. So thank you so much for watching.